Hello, I'm Dr. Ponticus, founder and CEO of Phenopolis. Phenopolis is a bioinformatics company based in London that delivers bespoke software solutions to researchers around the world. We are a team of scientists from University College London and Moorfields Eye Hospital and have been working together now for the last 3 years in building resources for researchers in genetics and ophthalmology. Myself, I've been working in genetics now for 11 years at leading bioinformatics centres such as the European Bioinformatics Institute, University of Cambridge and University College London. And one thing I've noticed working in these areas is that there's always a lack of good resources for geneticists who do not necessarily have strong coding experience to be able to explore and visualize their data that they've generated. The idea of Phenopolis came to me as I was working and running bioinformatics pipelines for various groups at University College London who do not necessarily have bioinformatics expertise. I wanted to give researchers the tools to make their data accessible and shareable with the wider research community, and for them to be able to integrate detailed clinical data using the human phenotype ontology in order for them to analyze gene to phenotype relationships. Because my team and I are ourselves researchers and scientists in academia with over 100 peer-reviewed publications, we really understand, I believe, better than most companies, what are the most important questions that we are trying to address, especially in the field of ophthalmology and genetics. Now let me talk you through some of our interesting products. So this is Phenopolis, our main flagship product, which is a database of genetic and phenotypic data that is browsable and facilitates visualization of analysis using to uncover gene phenotype relationships. So I'm going to log in as the admin user. And when I log in, I am presented with a screen which allows me to search by gene, phenotype, or patient, or look at a specific variant. So for instance here, I will go and look at a gene, ABCA4 gene. On the gene page, I'm given the name of the gene, I'm given the information about the full name, and also links to external websites along with the list of HPO terms, of which there are many in this case, linked to this gene in OMIM. Below is the table of variants contained in this gene, and this is a configurable and filterable table. It's configurable because we can remove the columns that we do not want to see, and it is filterable, so we can, for instance, filter by, let's say, allele frequency. This is the internal allele frequency in our database. So we can search for rare variants. Okay, maybe too rare. There you go. And then we can filter on other properties. For instance, the allele frequency in external databases. So we've got the Japanese inherited retinal dystrophy disease frequency. We've got Korean genome database allele frequency. We've got the HGVD, and we've got TOMO, which is also a Japanese cohort. We've got the Chinese consortium allele frequency, and we've got these very large normative databases, the Caviar, Genomad, on which we can filter on. We also are using CAD. So for instance, if we wanted to only look at variants with a CAD score more than 20, we would filter here. Now we have only 87 variants left. Now if we wanted to, for instance, see in which individuals these variants are, we could, just by hovering the mouse here, we can see that this variant is present in a person with a visual impairment. And by clicking on the individual, this will take us to the page for the individual and load all the variants in that person. Well, all the rare variants, homozygote rare variants, compound heterozygote rare variants, and rare variants. At the moment, we are using predefined thresholds, but we are planning on making those adjustable by the user. So we know about our ABCA4 variant. So let's have a look if that's in the rare variants of that individual, and we can do this by filtering here. And there you can see effectively this individual seems to have 2 rare variants in ABCA4. These do not seem to be reported in ClinVar. And now let's have a closer look. These are missense variants. One of which is one is a missense variant, the other one is intronic. So it is not clear whether this is the disease-causing mutation in this patient. The intronic variant is also seen in other patients, so it's not such a rare variant. It's also got an RS number. You can go and see more information about an actual variant now if you wanted, that intronic variant. And this is the variant page. At the top we've got information about the variant, its status. This is the genotypes tab, and this shows the genotypes of all patients within the database for that specific variant. If we wanted to find all genotypes, for instance, or patients which have a heterozygote genotype here, then we can sort, and we can see these are the only 2 patients which have such a variant. So this is our patient that we were looking at previously, and this is the other one. And we can also sort on the total depth, sequencing depth, and this shows us the allele depth for each allele, count basically of alleles. There's also information about the frequencies that we've seen before, as well as the consequences. So we've looked at the gene page, we've looked at the patient page, we've looked at the variant page. Now I need to show you how we can browse all the patients in the database. So this can be achieved by clicking on the My Patients button. Now there's more than 8,000 patients viewable at this point, so this page doesn't load immediately. As you can see here, we've got 8,627 patients. But we can also each row in this table shows you the patient ID, the gender, and the phenotype, and the number, and the candidate genes when available. These patients here have not had detailed and phenotypic information entered, which is why they have the all phenotype assigned. But say we were looking for a specific retinal phenotype, then we just have to type in retinal here, and this shows us all the patients with a retinal phenotype. It also shows us the candidate genes in those patients. Now, for instance, say we wanted to find out all the patients with a specific phenotype, then we can just go ahead and click on the phenotype, which will take us to the phenotype page. And as you can see here, we've got 5 individuals with this phenotype. Abnormality of retinal pigmentation. And we can get the results from our phenotype analysis and the results from our sequence kernel association test. So this just shows us which genes have a low p-value for this specific phenotype. So we've got MMA-DHC. So if we follow that gene, So this is an aciduria gene, cobalamin, okay, it's associated with these phenotypes. And this is potentially a candidate gene now for that patient's phenotype. Just shows you a bit of what is achievable using the Phenopolis interface. And we are working on developing more tools